It's been 6 months
since the discovery of COVID-19 virus, and it has spread rapidly across different countries. Like any other species, viruses also do evolve. Since last 6 months, coronavirus has evolved in different ways. So what happens in evolution is that there are genomes. They are basically the genetic material present in a cell which contains the information to build and maintain the organism. So a virus genome will make a virus, a human genome will make human. And there are mutation in these genomes. As we have heard that COVID-19 spread from bats, then to some animal, then to human. It's not perfectly clear yet. This is not uncommon because viruses do transmit from an animal to a human, like the MERS virus that originated from camel, the swine flu that originated from pigs, HIV from chimpanzees. But usually it is not that transmissible in humans. So there was some level of mutation in COVID-19 which made it so transmissible between humans. So some scientists were looking at the genome of the Sars-Cov-2. SARS-CoV-2 is the name of the virus that causes coronavirus disease. It stands for Severe Acute Respiratory Syndrome Coronavirus 2. The SARS-CoV-1 was the one that caused the outbreak in 2003, commonly known as SARS. Scientists were looking at the evolutionary relationship in viruses as they mutate. They looked at the genome of the virus right down to their level of genetic code. Since COVID-19 is a RNA virus, that means the genetic material stored inside is as ribonucleic acid. We know by basic high school biology that RNA has single-stranded structure, contrast to DNA which has double-stranded structure. This RNA string is made up of strings of nucleotide which form the basis of the RNA, and these nucleotides are arranged in certain code. This code tells us exactly what our genetic structure is going to be. The nucleotides are represented by letters A, U, G, C. These are the compounds that form the nucleotide. A stands for adenine, U for uracil, G for guanine, and C for cytosine. In DNA, instead of uracil, we have thymine. The arrangement of these codes in a particular manner determines which type of protein they are going to form. As I said, SARS-CoV-2 is a RNA virus, so it is one long strand of RNA which is made up of sequence of nucleotides, that is AUGC. So they might be arranged in A, G, U, A, G, and so on to form long sequences of letters, which is about 26 to 32,000 of these letters. If you compare it with human genome, we have about 3 billion of these letters arranged. And what happens is that the sequence of 3 consecutive nucleotides represented by these letters form a genetic code of a particular amino acid. And this string of amino acid combine together to make the protein that builds the virus. This amino acid forms 6 distinct types of proteins in SARS-CoV-2. So we get ORF1A protein, ORF1B protein, spike protein, E protein, M and N protein. We can represent this virus in a horizontal spectrum of the genome of the virus. So this horizontal line basically represents the protein component of the virus. In early January, researchers sequenced the first genome of the new coronavirus, which they isolated from a man who worked at the seafood market in Wuhan, China. This first genome became the baseline for scientists to track down the SARS-CoV-2 virus as it spread across the world. A cell infected by coronavirus releases millions of new viruses, all carrying copies of the original genome. As the cell copies that genome, it sometimes makes mistakes, usually just a single wrong letter. These typos are called mutation. As coronavirus spread from person to person, they randomly accumulate more mutations. This genome came from another early patient in Wuhan and was identical to the first case except for one mutation. The 186th letter of the RNA was U instead of C. When researchers compared several genomes from the Wuhan cluster of cases, they found only a few new mutations, suggesting that the different genomes descended from a recent common ancestor. Viruses accumulate new mutations at roughly regular rate, so the scientists were able to estimate that origin of the outbreak was in China sometime around November 2019. Outside of Wuhan, that same mutation in the 186th letter of RNA has been found in only one other sample, which was collected 7 weeks later and at 600 miles south of Guangzhou, China. The Guangzhou sample might be direct descent of the first Wuhan sample, or they might be viral cousins, sharing a common ancestors. During those 7 weeks, the Guangzhou lineage jumped from person to person and went through several generations of new viruses. And along the way, it developed two new mutations. Two more letters of RNA changed to U. Mutations will often change a gene without changing the protein it encodes. Like I said earlier, each amino acid is encoded by three genetic letters, but in many cases, a mutation to the third letter of trio will encode the same amino acid. These so-called silent mutations don't change the resulting protein. Non-silent mutations do change the protein sequence, and the Guangzhou sample of the coronavirus acquired two non-silent mutations. But proteins can be made up of hundreds of thousands of these amino acids. Changing a single amino acid often has no noticeable effect on the shape or how they work. As the months have passed and part of the coronavirus genome have gained many mutations, others have gained few or none at all. These striking variations may hold important clues to coronavirus biology. The parts of the genome that have accumulated many mutations are more flexible. They can tolerate changes to this genetic sequence without causing harm to the virus. The parts with few mutations are more brittle. Mutation in those parts may destroy the coronavirus by causing catastrophic changes in the proteins. Those essential regions may be especially good targets for attacking the virus with antiviral drugs. As mutations accumulate in coronavirus genomes, they allow scientists to track the spread of COVID-19 around the world. So in America, when the first man was tested positive for COVID-19, the genome of his virus contained three single-letter mutations, also found in the viruses in China. This allowed scientists to trace this man infection to its source, that is Wuhan. 5 weeks later, as the virus spread in different states, and scientists again took sample of an infected person and sequenced the genome of his coronavirus sample and found it shared the same distinct mutation found in the first case in Washington, while also bore down three additional mutations. That combination of old and new mutations suggested that the person did not acquire this coronavirus from someone who had recently arrived from another country. Instead, it was due to the spread of the same virus that arrived in the country earlier. By genetically sequencing over 2000 isolates of sample from different countries, scientists tracked how the novel coronavirus spread to different countries and how the virus evolved and geographically mutated in different areas. So in New York, the virus that scientists have sequenced show genetic links to COVID-19 virus in Europe. So as people traveled from US and Europe, the coronavirus carrying mutations that arose in Europe and US have been reintroduced in Asia. Over time, viruses can evolve into new strains. In other words, viral lineages that are significantly different from each other. A strain is a subtype of a virus characterized by different cell surface protein, eliciting a different immune response from other strain. A mutation is, however, a minor genetic error in genome sequence made during replication that doesn't fundamentally change the nature or the behavior of the virus. There are studies suggesting that this virus has evolved or split in two different strains or lineages called the L strain and S strain of the virus. So what geneticists think that there was an ancestral or old form of the virus called the S form, and this was the first form of the virus, and then due to genetic mutation in one of the S form, it mutated to another strain of the virus which is called the L form of the virus. So we have single ancestral form which is S strain, which is carried forward, but we also get a mutated form which is the L strain of the virus. And it is claimed by many prominent scientists around the world that this L form of the virus is more infectious and spreads more aggressively and is more deadlier than the S form of the virus. And it also spreads faster and causes symptoms that are much worse. The S strain was much common in the early outbreak of this virus in Wuhan, but later as it spread throughout different countries, it evolved into L strain. So in the last 2 weeks, we saw a spike in COVID-19 cases in Gujarat and Indore, and also increase in the mortality rate in these cities. Doctors believe that it may be due to this L strain virus vigorously spreading in these areas. Gujarat's Principal Secretary Health, Jayanti Ravi, had claimed that according to the information gathered by scientists, a different strain of the virus was behind the state's slower recovery rate.
हमारे यहाँ जो डिस्चार्ज होने की संख्या और जिसकी गति है थोड़ी कम है, और वैसे मृत्यु जो कोविड की चिकित्सा दरमियान जो मृत्यु होते हैं उनकी भी संख्या थोड़ी ज़्यादा है। तो इसकी जब हमने तज्ञों और जो निष्णात लोग हैं डॉक्टर्स उनसे चर्चा की तो बताया गया है कि दो प्रकार के इस वायरस के स्ट्रेन हैं, एक एस स्ट्रेन जो उतना घातक नहीं है और दूसरा है एक एल स्ट्रेन जिसकी ज़्यादा एक विरुलेंट जिसकी असर ज़्यादा होती है। तो इसकी भी एक अनुसंधान रिसर्च हमारे साइंटिस्ट कर रहे हैं और माना जा रहा है कि शायद इस स्ट्रेन गुजरात में है, इस वजह से ये थोड़ा इस तरह से मृत्यु के अंक भी बढ़ रहे हैं।
Doctors have sent more samples from Indore and Gujarat to the National Institute of Virology in Pune to do the further testing. Now the research about the L and S strain of the virus on human body is still under progress, and there is not yet proper research done to back this theory that one strain is more deadlier than the other one. Experts think that there is also definite sample bias for the L type, which was just sampled more and uniformly resulting in higher representation of the strain while testing. Recently, a new study suggested that there is another type of mutation in the virus that is concerning. Researchers say a new mutation called D614G in the spike protein is becoming more dominant across the world and is more infectious. The mutation in the spike protein can be more effective for the virus and make it more active because it is from the spike protein that viruses is able to infect human cells and replicate in our body. This 614G is just a name given by the researcher to the mutation. This report was published by the research done at the Los Alamos National Laboratory in the US. The preliminary findings from the lab reported hasn't been peer reviewed and formally published as of now. So more research is required before arriving at a conclusive proof. However, the authors note that their findings have important implications for COVID-19 transmission, pathogenesis and immune interventions. How does mutation of the virus affect the making of the vaccine? Well, it depends on the vaccine if it's able to prevent the infection from the mutated strain of the viruses as well. The mutation may affect or slow down the process of vaccine making, but as of now, the doctors believe that mutations likely won't interfere with the effectiveness of the COVID-19 vaccine. Because usually the scientists take into account the mutations of the viruses while developing a vaccine. And an older strain of virus will usually preserve enough features that it will provide immunity against a whole group of variants. When a vaccine is given, what we are trying to do is to simulate the infection, produce immune cells and antibody that will attack the infection when it comes in. We are given the form of infection to trick the body into making an antibody and cells to prevent the infection from happening. And this information about how to defeat the infection is stored so that when the real thing comes around, we are ready to go and kill the infection. Ideally, the vaccine will give you long-lasting protection, but we might end up with one that stops you from getting sick for a couple of months, like the seasonal flu vaccine, which protects you for about 6 months. So if the virus continues to be active, we may have to revaccinate ourselves after a couple of months depending on the duration of protection from the vaccine. And the short-term vaccine might be used temporarily while scientists work on a more durable one. So how long will it take for a vaccine to be developed? Truthfully, it takes decades to make a vaccine. Every stage of making a vaccine takes time, from sequencing the genome to isolating the virus to testing in cells to then testing in animals, going from mice to non-human primates. Also keeping in check the safety, efficacy and expanding the test in controlled environment before even asking for a license for manufacturing the vaccine. And then there is another challenge of providing this vaccine for the world population. Considering all this, Many experts believe and also reports suggest that with record-breaking time, we can get a coronavirus vaccine within 1 to 2 years. If against all odds, the tests provide positive result and everything happens as it's supposed to happen and doctors carry out extensive experiment, we may get a vaccine as early as 8 months. But it is also possible that we may never get a vaccine for COVID-19 because we haven't been able to find a vaccine for HIV for 30 years. So not able to find a vaccine for COVID-19 is also a possibility. Not to mention, many countries around the world are trying to develop their own version of this vaccine. And if they get somewhere, they're going to inoculate their own population first before they supply it to different countries. So it may take a while for the vaccine to reach us and is able to use it for the world population. I'm saying this not to discourage people, but to give them a realistic timeline on how long it may take for us to develop a vaccine. We see a lot of buzz in the media of vaccine being developed, but most of these companies are private and they have private interests. So these exciting headlines do help their stock. So it all emphasizes on how good the research and the funding of an organization is. After the SARS epidemic in 2003, the funding to many research organizations was cut down, and a lot of research money was taken out of the vaccine making once the vaccine was developed. So the further research didn't go on, which is disappointing because if people would have continued to fund this research, it would have been extremely helpful right now. So now in a way, we have to start from scratch with the vaccine making. When this pandemic dies down, we need to learn lessons from the past and need to fund science and research and make sure that when this thing happens again, we are more prepared. Mutation is a natural process. We will not be able to predict in which direction a species is headed. Here's something for everyone to think about. Since the beginning of this pandemic, we have been using hand sanitizer more frequently than we did before. So I wonder how many hand sanitizer resistant bacteria we are all accelerating the evolution of these days. Thank you for watching this video. Hope you enjoyed it.